So how does the protein able to communicate in different subunits, between the subunits? And so one way to think about this is if we think about, again, if we have a protein that has a certain molecular shape at the ligand binding site that can accommodate certain ligands. For instance, if this is the ligand and this is our protein, we can guess that, well, the molecular shape fits pretty well in this, and this, of course, as we mentioned, excludes different ligands from binding and helps in binding this particular ligand. So this is called a pre-organized structure of the ligand binding site. However, in reality, this ligand binding site might be very dynamic, and the molecular shape changes in solution. And so you might have, during certain periods, the protein may look, the ligand binding site may be very narrow or have a different shape, or it may be very wide and not look like the ligand at all. And so it can fluctuate between these different shapes, where this is the shape that promotes binding of the ligand into this ligand binding protein. And so this is the concept of thinking about proteins as very dynamic structures, so they move in solution. So typically when we see a crystal structure such as this one shown on the slide, we think of this as a snapshot, or we see this as a static picture. But really it's just a snapshot of the protein. And in the case of crystal structures, the protein has to be in a crystalline form, which is a really low energy state that really restricts the free motion of the protein. And in solution, in physiological conditions, the protein is not static like this. It's very dynamic. And so we can also use this dynamic nature of proteins to think about what are called conformational changes. So a conformational change, for instance, means that the shape of the protein or the arrangement of the amino acids may change ever so slightly within the protein at different times. And so we can think about these conformational changes in describing cooperativity for proteins as well, and how there can be communication between subunits, because if we look at the diagram on the right-hand side, we see a schematic of a simple a homodimeric protein that has the same binding site in between this blue cleft and this red cleft here. So these are color-coded, so our blue is very stable, so very rigid, doesn't move very much, doesn't have a lot of so-called conformational dynamics. Less stable than that are shown in green, and then very unstable, sort of kind of like flopping in the wind, are these red regions. And if the binding site is in between this blue and this red cleft, then this motion is going to have a lot of effect on how well the protein can bind the ligand. So if this motion decreases the affinity of the ligand, then what we can think of is if the ligand can bind this at a higher concentration, because this is a low affinity, let's say the ligand binds, and in this middle figure it binds and then it stabilizes this formerly red region. So it makes it have less movement within this binding site. And then that can cause sort of a tightening in the rest of the protein. So you'll see, for instance, this region in the middle that used to be green over here is now more stable. It's blue. And what it's doing is it's starting to restrict the motion of other regions within the peptide chain. So you may have less motion, for instance, now in the second site of this protein. And that is, in a way, how this is communicating. So this is by binding, it's becoming more rigid in the protein, decreasing the number of arrangements that this site can have, and therefore it's improving this prearrangement of the molecular structure, the molecular shape of the binding site, so that it can more easily accept the second ligand. And then that allows for a higher affinity binding of the second ligand to form the now 2 ligand bound protein. And so we have proof for this in hemoglobin. So there are 2 states within hemoglobin, and both of them have been crystallographically characterized. And so there is called the T, or the tense state, and the R, or the relaxed state. And so there are several structural differences between this. The most easily identifiable in the figure on the right is that there's a large central cavity in the tense state, whereas there's a much smaller central cavity in the relaxed state, or the R state. What is a little harder to notice is that in the tense state, the reason it's called the tense state is because the active site, or the region covering the ligand binding site, is more closed off. So it's almost inhibiting O2 binding to the heme, and is inhibiting, it's impeding the progress of O2 to bind to the heme here. Whereas in the relaxed state, this is released, and there's more space between these residues that may sterically inhibit O2 from entering the ligand binding site and the heme here. Okay, and in this case, we have this change in conformational dynamics, which switches between the T and the R state, and also, in effect, affects the way the heme can bind O2, how much access the O2 has to the heme, where the O2 actually binds to hemoglobin. And so this is, we can think of these T and this R state in these low and high affinity states that we were looking at for the conformational, in the concerted and sequential models for O2 binding to hemoglobin. So the tense state is the lower affinity. So as I said, this is the one where we can think of the K, or the P50 being 400 torr, whereas the relaxed state, the P50 is more like 4 torr. And we have these equilibria between tense states and relaxed states, and that may go through either a concerted model or a sequential model, shown on the right. But the important part, once again, is that O2 binding to a subunit induces changes in the other subunits to promote formation of more relaxed states. So as more O2 is bound to hemoglobin, it's more likely that other subunits within hemoglobin are going to be in the relaxed state, which have a higher affinity for O2, and in that way that's going to trigger more O2 binding to other sites on the hemoglobin. Now, whether that happens in an all or none fashion, such as proposed by the concerted mechanism, or in individual subunits, such as proposed for the sequential mechanism, we haven't discussed, but the important part, once again, is that there's able to be this communication between subunits based on whether O2 is bound or not, and that's through these T and R states. That's affecting the equilibrium between the T and R states of the subunits of hemoglobin.